Hello, we'll talk in this lecture about the development of the human embryo. The developmental history of a human embryo starts with the process of the gametogenesis, where sperms or ova are produced. The sperm fertilize the ova in a process called fertilization, and then a process of cleavage that takes place in which the formation the zygote is developed into a blastocyst. This process of cleavage followed by a process of gastrulation in which lasts for until week 4, then followed by organogenesis until birth. The process from cleavage until the organogenesis is the embryogenesis of the embryo. Fertilization. Fertilization basically consists of three phases. The sperm undergo a chromosome reaction in phase 1 and penetrates corona radiata, which is the region surrounding the ovum. In phase 2, the sperm penetrates zona pellucida, while in phase 3, the sperm and oocyte plasma membrane fuse. And you can see from the scanning electron microscope image how the sperms are surrounding the ovum and try to penetrate. Cleavage. Soon after the zygote formation and the completion of fertilization, the zygote undergo cleavage to have a 2-cell stage surrounded by zona pellucida, and then 4-cell stage, 8-cell stage, and so on for successive cleavage. The cleavage begins 12-hour post-fertilization. The zygote divides into 2 cells and so on by mitosis. The 46 chromosomes in zygotes equal the same number of chromosomes in every daughter cell produced. The 2-cell is transformed into 4-cell stage within 36 hours of fertilization, and 8-cell stage within 72 hours, and reaching Morula stage, which is a 16-cell stage. "72 hours post-fertilization, the embryo enters the uterus." In this figure, you can see the female reproductive system, and it consists of the uterus and the oviduct, which collect the ovum from the ovary. The secondary oocyte released collected by the oviduct waits for the sperm for fertilization until fusion of the female and male pronucleus. The zygotes enter the process of cleavage. You can see that the fertilization must occur on the first third of the oviduct. The cleavage soon starts until forming the morula stage. At this stage, the morula it is about to enter the uterus. Once the morula enter the uterus, it is converted by further development into the blastocyst. The blastocyst is characterized by an inner mass of cell and a ball of cells surrounding a huge cavity called the blastocyst cavity. Soon after that, the blastocyst is implanted into the uterine wall. Here you can see again the stages of cleavage up till the morula, and once the morula enters the uterus, after 3 or 4 days after fertilization, you can see the difference between the early blastocyst and the late blastocyst. The blastocyst is differentiated into embryoblast, which is the inner cell mass, and the blastocyst cavity, and a layer of cells called the trophoblast. The major difference between the late blastula and the early blastula is the degenerating zona pellucida in the early blastocyst. This is an enlarged image of the late blastocyst. You can see the inner cell mass and the trophoblast and the blastocyst cavity. In week 3 to 4, the embryo, especially in week 3, the embryo develop the amniotic cavity and the amnion around the embryo. And you can see a well-developed yolk sac. And the embryo now is connected to the uterus by the future umbilical cord, which is called connecting stalk. And it's surrounded, the whole embryo is surrounded by by a functional layer of uterus and the chorion, which constitute to the placenta. Early in week 4, the embryo enlarges and the yolk sac got a little bit consumed, and majorly the innervation and blood vessels to the uterus are increased. Soon in late week 4, the embryo, you can see that it's well developed, and you can see corneac villus in the placenta, and the umbilical cord is well developed, being contain the umbilical vein and umbilical arteries, and the yolk sac is still connected to the embryo late in the week 4. Embryo fetus. The embryonic period is from week 1 to week 8, while the fetal period begins on week 9 and goes until birth at week 38. The embryonic period is characterized by development of structure or organs. The fetal period is characterized by the growth of those structures. So again, this slide describes the fertilization up to the implantation. Actually, it's a more illustrative figure for the process of from fertilization to implantation. Going into details on the process of implantation, you can see in week 2 that on week 2, actually on day 6, the blastocyst tried to attach itself on the uterine wall. So you can see here that the inner cell mass will divide into two layers, the epiblast and the hypoblast, and starts to have the trophoblast to implant itself, the blastocyst, into the wall of the uterus. You will see that the embryo now is having two fluid-filled sacs, sac. One is called the yolk sac cavity, and the other is called the amniotic sac cavity. The yolk sac cavity is yellow, while the amniotic sac cavity is a blue one. The amniotic sac from the epiblast, yolk sac from the hypoblast, and these are the two sacs. Now the embryo is called bilaminar embryonic disc, and the area of contact, so because it consists of two discs. And this actually, the bilaminar embryonic disc will give rise to the whole embryo. So trophoblast or inner cell mass, as you can see from the figure, the organization and the location of each type of cells. You have an inner cell mass, which is the embryoblast, and you have trophoblast cells surrounding the inner cell mass. So the decision of cells to become either trophoblast or inner cell mass blastomeres is the first binary decision in our life. So actually, before this stage, it is a Morula stage. So it's a ball of cells starting from the 16 cell stage. It's the Morula stage. So in this slide, we will describe how these blastomeres in this cell mass, the Morula, will have a decision that some cells will be an inner cell mass and the other cells will be a trophoblast. What is the molecular mechanism controlling the fate of these cells? Prior to blastocyst formation, each early embryonic blastomere express both transcription factor CDX2 and OCT4. However, once the decision to become either trophoblast or ICM is made, the cells of these two regions express different genes. So generally, genes controlling the fate of each type of cells. So you can see that only the trophoblast cells synthesize the transcription factor CDX2, which downregulate OCT4 and NANUNC, which are two more transcription factors that, along with STAT3, characterize inner cell mass. So from the diagrammatic sketch, you can see that the morula have two fates. Either it goes, the cells, the blastomeres of the morula, either it will form the ICM or form the trophoblast. If it's forming the trophoblast, then it will have CDX2 that inhibit the OCT4 that promote the transformation into or the differentiation into ICM, while ICM is characterized by the expression of of STAT3 that's very unique for the ICM. So again, once cells are differentiated into trophoblast, it have CDX2, which downregulates two important genes, which is OCT4 and NANUNG. OCT4 and NANUNG promote the differentiation into ICM and soon after into the epiblast and the hypoblast. And the gene characterizing the ICM cells is STAT3. The activation of CDX2 gene in the outer cells is regulated by another protein which is called YAP protein. YAP is a cofactor for the transcription factor TID4. TID4 is found in the nuclei of both inner and outer cell compartment of the blastocyst, but is activated by YAP only in the outer compartment. So this is because the YAP protein can enter the nucleus in the outer cells and thereby allow TID4 to transcribe trophoblast specifying gene, which is CDX2, EOM's BSX1 and HAND1 genes. So you can see from the figure, once YAP activates TID4, it activates these three genes, EOMs and BSX1 and HAND1, which promote the formation of trophoectoderm. With the addition of the influence of CDX2, both the CDX2 activated by TID4 and YAP lead to the signal for the differentiation into trophoectoderm. In other words, for the blastomeres of the Morula stage to be differentiated into trophoblast or trophoectoderm, it needs a signal from CDX2, and CDX2 needs a signal from TID4, which needs a signal from YAP. One very important gene, OCT4, can downregulate CDX2, so they block the differentiation into trophoectoderm. So you can see here how the mechanism is working. So you can see here that YAP, once internalized into the nucleus in a blastomere, the nucleus of blastomere, and attached to TID4, it will promote the expression of CDX2. So these cells will fade into an inner cell mass. To describe the previous slide, actually the inner cells activate LATS protein through another protein, which is cell surface kinase, HIPPO. LATS is a kinase that phosphorylates the YAP protein. The phosphorylated YAP is not able to enter the nucleus and is subsequently degraded because it's not recruited inside the nucleus. Therefore, in inner cell mass, TID4 cannot function, so CDX2 remains untranscribed. CDX2 blocks the expression of OCT4, and OCT4 blocks the expression of CDX2. In this way, the two lineage become separated, the two lineage of trophoblast and inner cell mass. The expression of the three transcription factor characteristics of the inner cell mass, which are OCT4, STAT3, and NANUNG, is very critical for the formation of the embryo and for maintaining the pluripotency of the inner cell mass. In this respect, OCT4 is expressed first, and it is expressed in the morula as well as in the inner cell mass and the early epiblast. OCT4 blocks cells from taking on the decision to make a trophoblast cells. while nanong later on prevent ICM blastomeres from becoming hypoplast cells and stimulate blastomere self-renewal to form new cells in the epiplast. The activated, which is actually the phosphorylated form of the STAT3, also stimulate the self-renewal of the ICM blastomeres. Research on nanong is very important, and scientists are aiming to convert the normal human somatic cells into stem cell lines. The inner cell mass blastomeres are removed in a manner that lets them retain their expression of nanong-OCT4 and phosphorylated STAT3 protein. These cells divide and become embryonic stem cells. So in the process of doing experiment, they remove these cells and they term it embryonic stem cells. And you can see here from this synopsis that in early blastomeres you have two types of cell. In the stage early blastocytes E3.5, you have two types of cell, either trophoectoderm or inner cell mass. While started this inner cell mass become differentiated into primitive endoderm and epiblast. Soon after that, the primitive endoderm, after the process of implantation by stage E5.5, early post-implantation, this primitive endoderm is differentiated into parietal endoderm and visceral endoderm, while the epiblast just before gastrulation is differentiated into a definitive endoderm, a mesoderm, and ectoderm. Here you can see the blastocyst as the 7-day post-fertilization have been starting to implant inside the uterine lining. And you can see that the embryo now is consisting of epiblast, hypoblast, blastocele, and trophoblast, and is embedded into the uterine epithelium, which is the endometrium. Soon the embryo start to enlarge and start the cavity that appears, that is amniotic cavity. And you can see that parts of the trophoblast are proliferating into the uterine tissue. In day 9, the whole embryo have been, all of it is implanted inside the uterine wall, the uterine epithelium, the endometrium, and is now you can see clearly and you can differentiate between the amniotic cavity and the blastocele. And you can see that the lacuna starts to appear of the trophoblast lacuna. The embryo is now more developed inside the endometrium, and starts to get supply from the maternal side, and you can see that from the trophoblastic lacuna, the maternal blood supply. And you can see as well the embryonic epiblast and the amniotic cavity and the amniotic cells. If we wanted to describe how the bilaminar embryonic stage is transformed to trilaminar disc in week 3, so if you look this diagrammatic sketch, it's starting from the left side figure, you can see that the embryo now have two sacs, amniotic blue sac and the yolk yellow sac. This will take only this structure and will turn it 90 degree, and you can see from a 3D view, when you cut hypothetically, you can see where is the bilaminar disc occur. Actually, it's called bilaminar because it consists of two different types of cell, the epiblast and the hypoblast. The epiblast is the blue one and the hypoblast is the yellow one. So if we enlarge this bilaminar disc, you can see where is the epiblast and where is the hypoblast. And actually the hypoblast sits over the yolk sac, while in the epiblast it will be responsible for the formation of the primitive streak, which actually formed by the same process that occur in avian development we have discussed in the last lecture. Again, if you continue to look to the structure of the bilaminar disc, you can see that if you look from above, you can see where is the primitive streak. It starts posteriorly, represented by the tail, and you can see where is the right and left and head, that's the orientation. So further on, if you took a section through the primitive streak, you can see that the process of migration of cells from the epiblast into the cavity between the epiblast and hypoblast, similar to the process which was happening in the avian development. And these migrating cells actually constitute the endoderm and mesoderm, and this occurs on day 14 to 15. On day 16, start the mesoderm got internalized over the folds of the primitive streak, and the endoderm cells that have been involuted to the inside will arrange itself to form the definitive endoderm. In day 16, 18, the notochord starts to appear. So you can see here that now, after the cells have been internalized, and you can see that the primitive streak started to be elongated toward the head region marked by the red arrow. And the cells with the shaded red arrow can see that it is internalized over and move over the ridges of the primitive streak. And now the embryo, you can see that the embryonic disc is converted into 3 layers. That's why it's trilaminar, the ectoderm and the mesoderm and the endoderm. So the primitive node epiblast cells invaginate and migrate anteriorly with some endodermal cells. This road defining the body axis is formed, which actually is a future side of the vertebral column. You can see, if you took these two sections, you can see when you took a section at the level of the primitive streak, and you can see how the folds appearing and how the mesoderm are being invaginating mesodermal cells between the epi and the hypoblast. But if you take a section in front of the primitive node, you can see that as well there is a mesodermal cells between epiblast and hypoblast, but it is internalized from behind. So you can see that the notochord will be formed between the epiblast and the hypoblast. So basically, now the embryo is consisting of a trilaminar embryonic disc. At 17 days, the neurulation starts. Notochord signals the overlying ectoderm to form the neural tube. The neural plate to the neural groove, to the neural tube, they are pinched out of the body. So you can see from the figure, if you take a cross-sectional area, you can see the structure of the neural plate, the diagrammatic structure of the neural plate, and the surface got connected to the surface ectoderm, while lying beneath the neural plate, the notochord that signals to this overlaying tissue to form the neural tube. And you can see on the right figure the location of each type of tissue in relation to the other definite tissue. Of course, it's a color coding, so dark blue will form the neural tube, while light blue is the ectoderm, red is the mesoderm. Notochord appears between the mesoderm and below the neural plate, and of course you have endoderm and yolk sac. At 22, there is a closure of the neural tube begins at the end of the week 3 and could be completed by the end of week 4. So that's why folic acid is important for these steps. The embryos start to extend cranially, eventually brain, and caudally, eventually to form a spinal cord. The neural crest, which actually the neural crest is the lateral parts of the neural blades, lateral ectodermal cells are pulled along and form sensory nerve cells and other structure. So you can see here from the figure, if you took a section from a developed embryo, you can see that the neural tube have been already formed, which is covered totally by the surface ectoderm, and neural crest have been pinched off from the connection of the neural tube from the surface ectoderm, and the notochord is lying beneath. Soon after that, you can see that the mesoderm have been well differentiated into some mice, and soon will form the lateral plate mesoderm. And you can see that the neural crest is highly developed to later on will form the sensory nerve cells and other structure. If we look again about the mesodermal formation by week 3 and 4, you'll find that the mesoderm begins to differentiate lateral to the notochord at week 3. They extend cranially and caudally from head to tail or crown to rump. The mesoderm is divided into three major parts, same as what is happening in avian development and in amphibians development. Somites, which actually forms like around 40 pairs of body segments, which are actually repeating units like building blocks by end of week 4. You have intermediate mesoderm, which is just lateral to the somites, and you have lateral plate, which will soon be divided into splanchnic and somatic mesoderm, enclosing a cavity which will form in future coelom. So you can see here how mesoderm is differentiated. So you can see here that the folding of the future gut begins at week 4. So you can see here the embryonic disc, which is consisting of 3 layers, the upper ectoderm and lower endoderm and the middle mesoderm. So this sheet of cell, which is the trilaminar embryonic disc, started to fold to form the future gut digestive tube. Here you can see the total embryo at day 23 and 24 day embryo start to protrude into the amniotic cavity on top of the yolk sac. So you can see that somites, which already said that is around 40 pairs of somites, can be seen actually and visible across through the ectoderm. And you can see that head folds start to appear and tail fold as well. If you go diagrammatically as a section, you can see the layers of the neural tube, notochord, and the primitive gut. And as well, the primitive gut starts to make an opening in the anterior and opening in the posterior to form the future mouth and pharynx and the future anal canal. Going again to see the whole picture, you can see that the early blastocyst consists of trophoblast and the inner cell mass. Soon after that, the inner cell mass got differentiated into the epiblast and the hypoblast, and the two cavities arise, the amniotic cavity and the yolk sac. Soon after that, the embryo, as we said, start to protrude outside, and you can see how the neural plate is formed and the archintron, and how the heart start to develop, and how the future opening of the pharyngeal and mouth opening, and how the cloacal membrane that will contribute in the formation of the hind part of the digestive tube. As we said, on day 23 to 24, the embryo start to protrude into the amniotic cavity, and the lateral fold of the embryo will join ventrally. So you can see here the structure is well developed by day 24, and you can see that the mesoderm is well differentiated into the three main regions we have said before, and there is a lateral fold start to migrate ventrally and start to go around the yolk to form the lateral plates and to form the coelomic cavity to try to form the rest of the embryo. In day 28, the body takes the shape of a cylindrical human body plan, and this about half a centimeter in size. And you can see that the somite first part of the mesoderm have been differentiated into three regions, the same as amphibians and avian development. The somite is differentiated into dermatome, myotome, and sclerotome. And the kidney and gonad starts to appear as coming from the intermediate mesoderm, while the viscera serosa, which is the splanchnic mesoderm, derived from the splanchnic mesoderm, while the smooth muscles and the connective tissue of gut is derived from the splanchnic mesoderm as well. While the coelom will give the peritoneal cavity will give the coelom, which the cavity which surrounds the internal organ. You can see as well that neural tube, which came from the ectoderm, is well developed, and the epiderm, which come from the ectoderm as well, is well developed. And the gut lining, it's now complete tube, which is coming from the endoderm. The limb buds start to arise, and you can see that the parietal serosa coming from the somatic mesoderm is well developed. And you can see that the derm is coming from the somatic mesoderm, which will share in the structure of the skin, is well developed. If you take a section through the abdomen of the adult, it doesn't show a major difference, except that the knee spinal cord is a little bit smaller because you are in the caudal region of the embryo, and you can see that the vertebral column start to appear surrounding and protecting the spinal cord, and all of the other structure is actually the same as the section before. You can see that the trunk muscles are well developed now, and the ribs start to develop around the body. Now we'll talk about the eye formation and how tissue interaction during eye formation. You can see here that the brain, which is formed, is differentiated into three regions: prosencephalon and the mesencephalon and the rhombencephalon. And the prosencephalon, there is a region which is called the diencephalon. This diencephalon, if you look to its side, it will protrude to the outside to give the optic vesicle. This optic vesicle stimulate the outer lying ectoderm to become thickened to form the lens blackcoat. The lens blackcoat got invaginated to form the lens cup, with the invagination of the optic vesicle to form an optic cup. Soon after that, the lens cup got separated to form the lens vesicle, and the rest of ectoderm will cover over the lens vesicle and will form the future cornea. Now, the optic cup is differentiated into a neural retina and the pigmented retina connected by optic stalk. In this synopsis, you can see how the eye is formed through the developmental type of the mammalian embryo. The prospective ectoderm in early gastrula is differentiated into prospective neural plate and the prospective epidermis in the late gastrula. Starting from early neurula, the prospective optic cup is formed and the prospective lens ectoderm. These stages are being activated by the presence of a chorda mesoderm. Soon after that, in mid to late neurula, the optic vesicle gives the optic cup and the pigmented retina and neural retina, while the prospective lens ectoderm gives the lens blackcoat, lens cup, lens vesicle, and once there is activation of the neural retina, it will give lens. The lens vesicle as well activate the prospective cornea to be the functional cornea. Here we can see the major derivatives of Epiblast. Epiblast gives the three major tissues: ectoderm, mesoderm, and endoderm. From the mesoderm, notochord, somites, intermediate mesoderm, and lateral plate mesoderm arise. From the lateral mesoderm, somatic mesoderm and splanchnic mesoderm arise. From the ectoderm, a lot of structure develops, like epidermis, hair, nails, glands of skin, brain, and spinal cord, and of course neural crest and sensory organ and all the structures of blood vessels in head and neck. While from the notochord, it consists to be the nucleus of the invertebrate discs. Somites give the sclerotome, dermatome, and myotome, while the intermediate mesoderm gives the kidney and gonads. For the lateral plate mesoderm, the somatic mesoderm gives rise to the parietal serosa, dermis of the ventral region. The serosa actually a part of the epithelium. Connective tissue as well comes from the somatic mesoderm, connective tissue of limbs. While from the splanchnic, it will give the wall of the digestive and respiratory tract, except the epithelial lining, the visceral serosa, the heart, and the blood vessels. From the endoderm, it will give the epithelial lining the epithelial lining and the glands of digestive and respiratory tract. By this slide, we finish the mammalian development. We'll talk briefly on the types of monozygotic twins. So you have three major developmental fates that lead to twins. In first case, a splitting of the embryo at the two-cell stage occur before the formation of the trophoblast. Each twin by the end will have its own chorion and its own amnion. So you can see a twin having its own chorion and amnion. while in other cases splitting occur after trophoblast formation but before amnion formation, resulting in twins having individual amniotic sac but sharing the one chorion envelope. On the third developmental phase, you can see that the splitting alter the amnion formation, lead to a twin into one amniotic sac and a single chorion, and these are the twin that looks like identical twin. Now we'll talk briefly about stem cell therapy, the types of stem cells and where they come from. The first type is the tissue stem cells. So tissue stem cells allow us to develop, grow, heal, and replace worn tissue. They can come from many types of tissue. Most tissues have tissue stem cells, and they are important at all stages of life. The second type is embryonic stem cell. These cells are created from the inner cell mass of the blastocyst. So scientists can remove this inner cell mass and do research on laboratory on this type of cells. And cells can be collected and grown on plates in a laboratory. The third type is the induced pluripotent stem cells, in which cells from a person are genetically reprogrammed in laboratory, and these modified cells begin to gain the ability to self-renew and differentiate like embryonic cells, and soon after they can introduce into the patient for regeneration. So you can see here that they take the blastocyst, they culture it, they isolate the cell mass in a culture human embryonic and the culture human embryonic stem cells, and they can model and screen these stem cells for model for drug screening and toxicity assessment. Once they have specialized, they can able to differentiate. Actually, there is a lot of cells of stem cell can be differentiated. Specialized cells, if they succeed to differentiate these cells into different types, they can have they need availability of efficient differentiation protocol, and they need preclinical animal models as well as culturing and testing process are as required for human use. These cells can use as a cell therapy or a bioreactor, and successful engraftment of these cells at the maturation level and functionality of the cell, they need to be durable and feasible to have, for example, like a bioartificial kidney. So again here, what's a patient-specific stem cell therapy? So actually, you take a biopsy from the patient, you do nuclear transfer, you do nuclear programming, you develop into a blastocyst stage, you culture, you isolate, and differentiate into blood, nerve, heart, and muscles, and you transplant again to the patient. That's the main aim of a researcher working on stem cell therapy. Now we'll talk briefly on the evolutionary developmental biology. Evolution is a result of inherited changes in development. Modification of the embryonic or larval development can create new phenotypes that can then be selected. Darwin's concept of descent with modification explained both homologies and adaptation. The similarities of structure are due to common ancestry, homology, while the modification are due to natural selection, which is adaptation to environmental circumstances. The homology means that similarity between organisms or genes can be traced to descent from a common from a common ancestor. The ways of affecting evolutionary change through development at the level of the gene expression are the change in the location, heterophy, change in timing, heterochrony, and the change in amount, heterometry, and the change in kind, heterotypy. Evolution can occur through the thawing of the existing gene. Changes in the location of the gene expression can explain the origins of novel structures such as feathers, turtle shells, and dog feet. Changes in the timing of gene expression have been important in the formation of whale flippers, lymphs through the animal kingdom. Epigenetic inheritance system, example, inherited pattern of DNA methylation. So when a gene is heavily methylated, it will be switched off. It's the same as if the gene is genetically mutated because it has no function. Evolutionary developmental biology is able to show how small genetic or epigenetic change can generate large phenotypic change and enable the production of new anatomical structure, as we can see from this figure. In this figure, we have a UPX protein phylogeny through the arthropod. So UPX protein is responsible for the appearance of legs in the abdomen. So once it is mutated in some group of animals, mutation lead to the formation of a string of any alanine, which is called the polyalanine in the protein. Once this mutation starts, you can see all the descendants, which is actually insect, fly, mosquito, butterfly, and moth and beetles. They have this mutation of a string of polyalanine. That means this mutation lead for these organisms to have abdomen without legs. Although the protein shows similarity along the arthropods, it differs in the class of insecta, which shows no on the part of the polyalanine stretch as a result of the mutation. And of course, the one who is responsible for the theory of evolution is Darwin's. Please don't forget to subscribe to get all the updates of the lectures. Thank you.